Hello, everyone. Thank you so much for being here for tonight's Architects of the Brain. Tonight is a fascinating panel. I was so thrilled to get this one because some scientists, their passions lie on the bottom of the sea, and some are focused on space, but we have four people here today who are obsessed with that beautiful space between your ears. So we've heard a lot about the brain in headlines in recent months. President Obama making one initiative to map the human mind. Of course, there's one going on in Europe as well, where they think if they have a decade and €1 billion, they might be able to actually literally create a model of the human brain in a computer. But is this wise? Is this even possible in any way? What are the benefits of trying? And if they are able to actually create a very, very smart machine and it reaches self-consciousness, will it send Arnold Schwarzenegger to kill us? All valid questions for our panel, so let's just get to it. We have so much to talk about tonight, so help me welcome our four participants tonight. Our first is a leading expert in white matter, everything that is not a neuron in your brain. He is a developmental neurobiologist, author of The Other Brain. Please welcome Douglas Fields. You got a whoop. He's got a fan club. Also joining us, one of the world's leading neuroscientists investigating synapse structure and function. She's a professor in the section of neurobiology at the Center for Learning and Memory at the University of Texas, Austin. Hook 'em horns. Please welcome Kristen Harris.
Hook 'em horns.
Hook 'em horns. Our next participant traveled the farthest. He is a professor of cognitive robotics at Imperial College London. He's been focusing on the brain, relationship between cognition and consciousness for about a decade now. He also helped start the Existential Risk Institute at Cambridge. Join me in welcoming Murray Shanahan. And our final participant is a computer scientist and a philosopher and editor-in-chief of Minds and Machines, a visiting associate professor of philosophy at Carnegie Mellon. Please welcome Greg Wheeler. Gregory. Thank you so much for being here. And I will remind you, we want this to be a crackling conversation. So I encourage much interruption and shouting over each other. And we're going to use the narrative device of building a brain tonight to take us through this next 90 minutes and to kind of understand where we stand in the development of an artificial machine that has the power of the human mind. We want to know whether they agree on whether it's possible, whether we should even try. So each of them are going to contribute to this. So let's start this out just by setting the table with what a huge challenge just this very idea is. So Chris, why don't you start us out? What are the challenges of doing what we're talking about tonight?
Well, one of the biggest challenges is that we actually don't know where every connection occurs in the brain. And in fact, we know precious little about where every connection occurs in the brain. And so one of the biggest challenges is to develop the tools to be able to actually go in and map how the brain is organized as a starting point. If you're going to build a brain, you actually have to know how it's put together in the first place. So I'd say that's the first big challenge, is to overcome how little we know in terms of the overall detailed structure of the brain. And we're going to get a chance to see what that looks like in its nanoscale.
Yeah, you've got some great animation to show us as well. What do you think, Doug? What are the big challenges?
Well, it's an enormous challenge to try and understand the brain and make a model of it. You know, the brain has always been the most fascinating mystery, I think, for mankind. It's been called the most complex structure in the universe. And so we've always tended to try and understand it in terms of analogies to the most current technology of the time. And that could have been mechanics or hydraulics. And today, we tend to think about the brain in terms of an electrical computer. And it's humbling to realize that the brain may not work like that at all. And I'm mindful of Leonardo da Vinci. He made meticulous, precise observations of anatomy and drew some of the best anatomical drawings we have. But when it came to the brain, he got it all wrong. These are the ventricles. They don't look like that at all. And he's got the eyeballs wired up to the ventricles through the optic nerves. Now, if you just look at the tissue, you would see it doesn't look like that at all. But he was blinded, his perception was blinded by his preconceptions. So he had this idea about how the brain worked, and then he that blinded him to what he was actually seeing. Later in life, he did make some corrections and got a little bit more accurate. But I think that's a little bit humbling as we begin tonight trying to think how to
Well, he wasn't the last scientist to be wrong about it. I mean, it's
No.
just one in a long chain. But Greg, it's not just anatomy and physiology, it's function, right? It's what it actually does and how it does it.
That's right. I mean, we're principally interested in the brain because of the things it does for us. We have beliefs, we have desires, we want to do things in the world. And the thought is, so we have mathematical theories of decision-making, for instance, how to make good and rational decisions. We go out and we look and see what people do. They don't do what the normative theory often tells you to do. So the question is, how do people make good decisions? And as we'll see through the discussion tonight, it seems that one of the ingredients that we need are constraints. We're going to see how complicated the brain is very shortly. And one of the themes that we're going to return to is that it seems necessary to constrain the brain in order for it to do things that are familiar to us. So I think a challenge is to see how far we can come in understanding what some of these physical constraints are to everyday actions that we have and decisions that we make.
And Murray, your reaction when you hear these ideas about mapping, creating these models, from your discipline, is it crazy in your mind, or you get excited by the possibilities?
Oh, I think it's a very exciting possibility, because although I think it's probably is right to say that the brain is not really like a computer, but nevertheless we can build computers that are like brains, which is a very different kind of thing. So we can use computers as a tool for simulating brains and trying to understand them better, and it's that challenge that really excites me.
Okay, well, let's start with the basics. Let's get a little schooling on what exactly is happening upstairs. So, Kristen, how does a neuron work? Let's start there.
What you're looking at here is a neuron. It happens to be a pyramidal cell. It's one of probably several hundred different types of neurons that are found in your brain. It's considered a canonical cell, so we often refer to this type of cell when we're looking at a neuron in the brain. And this is how it looked when Golgi and Cajal and Nansen first began studying the brain around about the turn of the last centuries. And they began to really try to understand how neurons worked from images, learning by looking, in fact, as how things were connected to each other. And what you see here is the neuron cell body, which is sort of the engine for producing a lot of the organelles and the proteins that are going to be maintaining this vast network of dendrites which receive inputs from all over the brain. And so this one cell might receive as many as 20, 30,000 synapses from different cells in the brain. In fact, most of those are from different cells. We've actually mapped axons passing through. So now the axon, you see an axonal bouton, and then you see these little tiny protrusions that come off the surface of these dendrites that are referred to as dendritic spines. Now, the way these neurons communicate with each other is through the axon receiving enough input to actually fire onto its neighbor and transmit across something called the synapse a signal that tells the downstream cell something happened upstream, you need to send this message on, or not. So there's a lot of integration that goes on. So the synapse on one of these dendritic spines now is shown to you at a much higher scale. You see a little bar down there at the bottom, that's 500 nanometers. It's very tiny structures in the brain, and they're very compact within the brain, and you're viewing them now as I view them in the electron microscope. And this has been, a big leap from going from the light microscope to electron microscope, because now we can actually watch at the precise level how the substructure of the axon and the synapse and the spine and its neighbor, which my colleague to my left will spend a bit of time talking about more, the glia, interact to refine and shape the communications that occur through the synapses in your brain. And there are billions and billions and billions of synapses in your brain. So we're going to show you a little movie, and we call it a cruise through this neuropill. And the neuropill is this felt work structure of the dendrites and their axons and their synapses and their glia, all knitted together within this complex goo that you see when you see a brain spill out, on somebody's gotten shot in the head or something like that, right? That complex goo is this network of synapses that are occurring in your brain. So what we've done in the next sort of 4-minute movie clip is to allow you to see how we, as biologists, try to build a brain and try to, in 3D, take a real brain. This happens to be a brain from a rat, in which we've done some experiments on and tried to understand how these synapses change their structure as the animal learns something. And this is from the hippocampus, a brain region that we know. If you lose the hippocampus, you will not be able to get another new memory. All your old ones will be intact, your new ones will not happen. So as we go through the cruise, this neuropill, you're going to see a central dendrite. The postsynaptic density in red is where those synapses occur, and there'll be some organelles, a mitochondrion will come up in purple, and we'll see some others too, and the axons will be in green, the spines will be in yellow, and the synapses will come up in sort of a copper color. And then we will have the structure that we'll spend more time on later, the Astroglia will come in to dance with the rest of this neuropill. So we chose this volume to be a volume that was sufficient to surround this dendrite at the reach of its longest spine. So the first question one can ask is, gee, you have this dendrite, has all these little spines sticking out, how many synapses can it make? And of those synapses, of those axons that come within reach of those spines, what fraction of them actually make a synapse? So now coming in to dance with this dendrite are all the axons in that little tiny volume of neuropill, and you're going to find out how small it is in a moment, that actually make a synapse on that central dendrite. And two of us, Joe Spacek and I, actually manually traced every one of those through 200 serial sections to identify where the synapses occurred. So coming to join them for a moment here is another third partner in the neuropill, a critical partner, and this is the glia, and they interdigitate also as part of this nice felt that is all communicating to make your brain work at this moment. At this moment, your brain is changing, actually, in fact. Some of those synapses are going away and some of them are getting bigger. Because as a mature person, you can't support all the synapses you could possibly have. So now we've jumped into the way I see this tissue when it comes off of the electron microscope. These are the electron micrographs. We're zooming in and out, and all of those structures are structures that I traced with a computer-assisted program and colorized them, named them, so that there's some ground truth for these guys who are going to model the brain to know which structures are next to each other and how they interact with each other. Also inside, we have, and now we're going to get to see them in 3D, we're zooming in on that dendrite, and you're going to see just how complex the substructure of these dendrites and their axons are to support these synapses. So the first, you're seeing now the dendrite, and in red are those synapses on those spines. We've made this translucent so you can see some of the 3D structure inside. So the challenge is not just how many connections there are, but how are they supported by these structures that build those synapses. So there's 3 mitochondria that are down the central shaft of that dendrite. And those little white spheres represent the places where protein synthesis was occurring at the time that this tissue was fixed and made. They're called polyribosomes. The blue structures coming in are the cytoskeleton that provides support and the railroad along which all these organelles and proteins get transported. And then now this network, this spider-like network of smooth endoplasmic reticulum is what provides support for regulating ions like calcium, critical ions in the brain, regulating them and also allowing other membrane-bound proteins to move up and down that dendrite to the right place. So we saw a little bit of that felt work neuropill. We saw the dendrite and its axons. Now coming to join the dance are all the other dendrites that surrounded that same central dendrite that we thought was so important, right? Well, these guys are also competing for the axons. And those few axons that I showed you before only synapsed with that central dendrite. Now, the rest of these axons were within reach, but didn't make it. Didn't make a synapse with the central one, but they're synapsing with these other dendrites that are in this volume. And so it's this kind of knowledge that we need to have before we can really have a realistic idea of exactly how your brain is working. And as we go through this, I'm going to tell you at the end here that there are 500 synapses in this volume. 500. This volume is the size of a single red blood cell. It is less than 200 cubic microns. So all of that volume of brain that took us years to reconstruct manually using a computer and putting into this value is so tiny that if we were going to map the entire human brain at that level, we'd have to repeat what I just showed you 8 trillion times. So this is why when Bill asked me, what do I see as the big challenge of constructing a brain, the big challenge is giving me, the scientist, the tools to do that kind of level of analysis and work at a much faster level. And I think that one of the grand challenges of the Brain Initiative, which is really what it was built for, was to both be able to marry those of us who are looking at this detailed structure to those who are looking at the detailed function of each of these synapses within the brain. And I think it's a really wonderful time now for this Brain Initiative to be happening because the tools are getting better and better.
We were talking earlier about the differences between mapping that and mapping the human genome or something, but and expound on the idea that it's changing all the time. It is a moving target. It's trying to sculpt something that is shape-shifting.
It's absolutely shape-shifting. So we might get one human brain in, 1,000 of our lifetimes all working on it together at this level with the tools in hand. But your brain and your brain and your brain are not all exactly the same. And so trying to figure out what the statistical brain is so that you can say, well, gee, Bill's brain is normal and mine is a little bit off. We're really going to have to. I wouldn't go that far. We're going to have to not only get this kind of mapping and this kind of level of knowledge about one brain, which is our current mission, we're going to have to figure out what are the principles that can be multiplied and thought of as principles that apply to all brains, and that also then what is a deviation from a normal principle. And I think this is another level of really huge challenge, is deciding, where you draw the line as to something has become ill and it needs fixing versus, that's a genius in the making.
Right. Let's. This next bit of animation helps put this in perspective, just the problem here. So how many, put it in terms of computer power, say like laptop per neuron ratio. How many?
Well, so, I think we all kind of guess that a single one of those brain cells probably has the power of your whole laptop. And there's hundreds of billions of cells in the brain. And so I guess the first animation is to give you a sense of what 100 billion
we figured
laptops
Central Park might
like.
just start.
So making room for all of that computing power in a brain is really also one of the challenges that evolution has done that
but Murray,
computer scientists haven't yet.
Chime in here on just the technology that exists today in terms of just showing us that, how you go from a cross section in a dish to animating that.
Yeah, well, what we really want to do is to be able to scale up the kind of thing that Kristen and her postdocs have done and really speed it up dramatically. So the first thing that there's sort of several steps to this. The first thing that you want to do is you want to be able to make these slices very, very rapidly. So you want to be able to automate the business of taking a little bit of neural tissue and making the many, many slices in an automatic way. So some people have been working on machines that will do that and will lay them out on a tape. Then the next thing that you need to do is you then need to use your electron microscope to scan in each of these little tiny slices, so you get a picture like the kind that you saw in Kristen's video, where you have one sort of snapshot, one slice through the brain. But you've got to scan millions and millions of those, so you really need to dramatically speed up that process as well. And then the next part that you have to do is that you then have to do a kind of reconstruction, because then what you've got is like a stack of plates, where each one of those plates is one slice through this little bit of tissue. And if you can imagine this stack of plates. If you've got an axon which is passing through the stack of plates, when you look at one of the plates, one of these electron microscopy images, what you see is a cross section. So you might see a little circle in that top bit of the stack. And then you look at the next bit of the stack down, and then you'll see the same kind of circle, and maybe shifted a little bit further along. And as you can imagine, each of these cross sections is going to show you lots and lots of little circles going down. And somehow or another, so what Kristen did painstakingly was to color each of those little circles in order to figure out where that one axon went down through all of that stack of plates. But there's no way that you can do an entire brain like that. So that whole process needs to be really, really dramatically speeded up. And the only way to do that is to automate the process with sophisticated computer vision techniques.
And Greg, you can speak to this too, because a lot of the modeling is done using mathematical formulas that were invented in the 1950s. So not only do we need much better computers, we need a new algorithm system.
Yes, and even the underlying mathematics. I mean, there's an old joke in statistics departments that, the bell-shaped curve, this Gaussian distribution of error that it's normally distributed. This and stationarity, these are things invented for undergraduates. In real life, errors aren't necessarily distributed normally, and we're describing a process here, a time series data that's not stationary, it's highly non-stationary. So all the tools that we're using, most of them anyway, are built with these fundamental assumptions, but we're using them in an exploratory fashion. So we're trying to bootstrap with these old tools, knowing that they're introducing biases. We just don't know how grand they are. So this was the point that Kristen was raising about the techniques that we need. So in addition to the computational machinery and improvements in vision, there's fundamental advances we need to make just in the underlying mathematics of how to explain and measure processes of this kind. So I think the developments and the advancements that we'll see will have impact well beyond the topic that we're discussing here.
Right. And so the neuron, just the beginning, as we start to pull out a little bit now, the connectome. So Kristen, Doug, tell us what's happening here.
This is sort of a coloration, as I understand, of axonal connections in a human brain. And I think Doug is maybe even more expert on this particular animation. And
well, I think the point of this animation is that one of the goals is mapping all the connections in the brain, that the brain works through networks, very complicated networks. And so that's one of the main goals, but it's an immense problem. In addition to the technical problem, you have to be careful what you wish for sometimes. So let's say we do get all the billions of connections. Can we understand it? Or you're back where you started? I mean, we have this problem in gene analysis. We look, and others in the lab look at thousands of genes changing at once, and we can't get our head around it. Well, we have that problem.
Right. And so this billion-euro effort being led by Henry Markham in Europe, is it taking these complexities into account? I mean, this is for anybody can jump in on this.
not yet.
Not yet.
But I think that's the vision. And I think that's the vision also of the BRAIN initiative in this country. And maybe is even an answer to the European, and we could go for a few billion ourselves in this country. But I think that we're not there yet, and I know that, Henry's working very hard to get a sense of how the columns of the brain at the light microscopic level are interconnected. And so this is where the idea of how does the brain repeat itself? So that if I were to do maybe only 2,000 of these and to sample it just properly in the brain, I could get some sense of, well, maybe I don't have to do all 8 trillion reconstructions, but that it could be scaled down because there are places that are similar, and you start to learn the principles. And so I think right now where the Human Brain Project in Europe stands is to try to get an idea of how the columns of the brain, the cells in the columns of the brain repeat themselves, and whether or not the anatomy and the function can be repeated, so that would actually help to guide those of us who are looking at the synaptic level itself, to guide us as to, well, gee, where should we go next to do this kind of painstaking work as we even start to build the new tools. And I think, some of the vision of the BRAIN initiative is, if you look at the curve of advancement, it's kind of a linear, not a very steep slope. My colleague Mi-Young Chun at the Kavli was telling us about this today, that we're really at a point, if we can bring more minds that are thinking from engineering and from physics points of view together, if we can bring us all together to really share our different points of view, that slope may start to go more vertical. And I think that part of this effort all across the world to understand the principles from many different viewpoints, from physical viewpoints, from engineering viewpoints, that the time is to really share and not only to compete for new resources to study our own little favorite part of the brain.
So that's the perspective from someone focused on neurons. Doug Fields, you focus on the other brain, as your book is. So what was your reaction when you heard about the idea to map?
Well, I think it's a fantastic idea, a wonderful idea, but my first reaction was that, well, the map is going to look a lot like those early nautical maps that left out many of the continents and had a lot of detail on the home ports, because the map is leaving out half the brain, and I'll talk about that later.
Okay. What are glia? Give us a schooling.
Well, we all know about neurons, but only 15% of the cells in the brain are neurons. And you're looking now, just as Kristen showed, electron micrograph looking into the brain. This is from Mark Ellisman's lab in UC San Diego. And only 15% of the cells are neurons. You can see blood vessels, and the majority of cells are cells that don't make electrical impulses, and these are called glia. The word means glue. At this scale, you can't even see synapses. So if you're trying to understand communication in the brain only through connecting synapses, I would say you're overlooking the obvious. There has to be a lot of other communication going on in the brain, communication that may not be electrical and is probably not synaptic.
What is it then?
Well, we can get into that.
Okay. Sorry.
Right here we show blood vessels. I just make the point that glia control blood vessels. Your brain can't be on 100% all the time. Your brain takes 20% of the energy. It's only 5% of the mass. So local blood flow is turned on to different regions of your brain, and this is how we can see the brain working in these functional MRIs. So as I'm sitting here looking around the room, different parts of my brain are getting spurts of blood to turn on those circuits. The glial cells are very much involved in doing that in response to neuronal activity and demand. But let's go to the next one. And,
Okay.
The point being, here's a neuron. And with all our fascination, and I love neurons and I love synapses, the synapses are shown here on the dendritic spines. And if you counted the number of synapses in the human cortex, it would take you 32 million years, counting one per second. Good project for one of your surly students.
I need many more than one.
But the point I'd like to make here is, while we're focused on the neuron, the eye sees what the mind is prepared to comprehend, we're ignoring what's in the background. And the textbook image of neurons always leaves this black. And what's in the black are glial cells. And these are the cells that communicate without using electricity, so they've been kind of ignored.
We know that it's apocryphal that we only use 10% of our brains. We use all of it a lot of times. But part of that old saw is that Einstein then used 12% or whatever it is. But what is true is that he did have a different brain.
He did. When Marion Diamond looked at his brain to find the cellular basis for genius, she could find no difference in the number of neurons or the size of the neurons in the cerebral cortex involved with mathematical ability and imagination. But she found that he had more glial cells than normal. So that kind of really focused attention on what could glia be doing. More recent research has shown that his glial cells were different. He had a lot of very large cells. They were bigger, they were more complicated. And these glial cells, called astrocytes, there are some that are unique to humans. They're much different from rodents. They're 20 times larger volume. And so when these cells were taken from humans and put into mice by Nedergaard and Goldman, these mice were not geniuses, but they were brilliant. They run through mazes much faster. So they have something involved. So what do glia do? How do they communicate if they don't have electricity? Here I have some astrocytes in my lab and culture. I've put in a dye into these cells that allows you to see them communicating, not with electricity, but by waves of calcium ions. So this dye fluoresces when calcium enters the cell.
And what happens if there is a malfunction in the glia? How does that manifest itself?
Well, before malfunction, maybe I should, before I talk about that, maybe tell a little bit about glia, the kinds of glia. We talked about glia. It's like there are many kinds of glia. And they're into 4 major categories. Glia called astrocytes, and we'll show some of the background. They're nothing like neurons. They surround synapses. They release neurotransmitters. They can control synaptic transmission between neurons. They can pick up the signals from neurons communicating at synapses, control that synapse, communicate among themselves by releasing neurotransmitters, sending messages through glial networks, not neural networks, and control glia and other parts of the brain. So those are some of the things that astrocytes do, and it's another level of function and control on the brain. There are many kinds of glia. Another kind of glia are microglia. Microglia are involved, they're the immune cells of the brain. So in any kind of brain repair, glial cells called microglia are involved. But we've recently learned that glial cells, called these same cells in your normal brain, are involved in unplugging synapses. So they make and break these connections we're talking about. That's another difference, that microglia move. They're moving like worms through garden soil in your brain right now, sensing all these synapses, and based on the activity in those synapses, plucking some of them off and rewiring the brain. This is all new research. Another kind of glial cell is called an oligodendrocyte. It's a myelinating glial cell. People know about myelin. It's the electrical insulation wrapped around axons that allows for high-speed information processing. It speeds impulse propagation through axons 50 times. So the difference between a myelinated and an unmyelinated axon would be like a race between somebody walking a mile an hour and somebody driving by 50 miles an hour. Oligodendrocytes are why we have a brain, I think, in evolutionary function, because oligodendrocytes developed only with vertebrates. And when you think about it, there's a huge difference between cognitive ability of worms and slugs and snails and people and dogs and dinosaurs and fish that have backbones. The difference is they have oligodendrocytes. That allowed us to send impulses long distances. These invertebrates have to have neurons stashed wherever they need them, like next to the mouth if they're going to move the mouth. But we were able, and through evolution, we were able to form a brain because the cells could come together and yet communicate long distances.
Wow. But getting back to the, yeah, exactly. A layman's term. But getting back to the malady, the manifestation of bad glia,
Sure.
Is that like ALS or?
Sure, ALS and many of the diseases we used to think of as neuronal diseases, ALS, epilepsy, Parkinson's, all involve glia. Even psychiatric illnesses, depression, schizophrenia, all involve glia, in part by regulating synaptic transmission. Stephen Hawking, ALS, originally thought to be involved only with motor neurons dying, but it involves the astrocytes as well as the oligodendrocytes. So many disorders. HIV causes dementia, and as people are living longer with retroviral drugs, they're suffering dementia, increasing numbers. HIV doesn't infect neurons, it infects glia, only astrocytes and microglia. Alzheimer's is associated with this accumulation of beta amyloid. Microglia eat beta amyloid, so if they don't remove it, plaques can form, and microglia become senescent and can lead to Alzheimer's. So I could go on and on with this, but I would also, I'll stop there.
Okay,
yeah.
Well.
I could go on.
Yeah, give him a hand. I mean, but in the battle royale between glia and neurons, Doug has the upper hand, Kristen. You have a chance to come back.
Oh, no. I must say that I'm also a great studier and lover of Astroglia. And in fact, our lab was one of the first to show that not all synapses have Astroglia at them in the hippocampus in particular, but also in the cortex. And it turns out that those synapses that have astroglia even just touching a part of their dendritic spine, those synapses will be 50% bigger than a neighbor that doesn't have them. So this isn't a battle. This is a partnership.
A symphony, right.
In fact, we all consider the synapse to be now basically the tripartite synapse. There's the axon, there's the postsynaptic partner, and there's the perisynaptic astroglia. But what Doug has pointed out is it's now really becoming clear that it's a quadpartite, because the microglia is always there deciding whether or not that synapse is going to get to stay on board. So we really He's making really a beautiful point that if we were even to get every single connection in the brain right, we really have to understand how it's functioning and how it's functioning in relationship to the other structures that are part of the brain.
So Doug, let's go back to our World Science Festival brain project. We're building this. What would you do? You wouldn't think about it like a computer.
Well, first, first of all, the brain's not a computer, and electrical computers are not, digital computers are not going to be digital in a few years. They'll be quantum computers. So it's not rocket science. It's brain science. So let's begin with what the brain is. The brain is made of cells. And what are cells? Cells are bags of seawater. So over the course of evolution, nature has come up with these bizarre shapes, neurons and glia, coming together to make this brain. Now, the reason I put this out is, make this point, is also, as we're amazed at how the brain works, consider the engineering problem that nature had. But the fact that there are no wires, there are no hard drives, there's no digital code, there are no programs. This reality that the brain is made out of bags of seawater, 1 billion bags of seawater, puts some constraints on how the brain works. If you approach the brain with a certain framework, you may actually miss how it works. So, for example, neurons, axons don't send electrons down them like electrons through copper wires. They communicate by having pores that allow dissolved salts to go in and out. Potassium chloride goes out, sodium goes in. And what happens when it gets down to the synapse? How does it communicate? Squirts out bits of cytoplasm. The point is that you have, the rest leaked into my pocket. The point is that you have these physical constraints. Now this really affects how the brain works. So if I give an example, you go to the doctor, he does this knee-jerk reflex. What's that about? That's about, as we walk, if we were to stub our toe, we would have to swing our leg forward in a split second to prevent us from falling on our face. So let's design a circuit that does that. Well, the problem is, I wish it was a laser beam or Wi-Fi, but it's not. It's a squirt gun. So what did nature do? Nature took the neuron in your toe and stretched it out till it was 3 feet long, and it goes up into your spinal cord. Absurd. Humongous neuron, 3 feet long. The biggest cells in your body are neurons. Once it gets to the neuron, spinal cord, it squirts onto the motor neuron. Another ginormous cell comes down and does to contract the muscle. The reason we have that is we have only enough time for one synapse in that whole circuit, because this takes a long time. And glia clean up the mess so you can send another synapse. So we don't communicate at megahertz. Axons don't communicate at megahertz rates. They communicate at rates of only a few hundred a second. The point is, in order for the brain to work, there must be something more than just connections. And there are. There are things that couple neurons together. There are brain waves, oscillations in the brain of different frequencies that couple neurons together and make them fire even though they're not connected, something that we need to consider if we're going to build a brain. And I would also say that many of us believe that glia are important in that coupling, because glia can encompass in the human brain, astrocytes, 2 million synapses. So they can control local domains of synapses or even populations of neurons. So the glia could be like a conductor in an orchestra, making these groups work together and these groups work together, and this would lead to higher level cognitive function. So, yeah.
I like it. So Murray, you're a cognitive roboticist, and so if Doug's model brain involves water balloons and squirt guns, apparently. If we could do what people want to do, what kind of technology do we need, do you think?
Well, I'm certainly not going to argue with Doug because he's got a water pistol.
He's got a gun.
Well, so to think about that, let's take a look at the first video. But don't. Oh, I was going to say don't run it yet. But we'll run it again. So this is a really beautiful video which shows the kind of thing that we would like to be able to simulate on a computer. And this video is from Misha Aren's lab at the Howard Hughes Medical Center. And what it is it's the brain of the larva of a zebrafish. And it's showing the activity inside this brain, and this is in vivo. So this larva fish is this zebrafish is alive, and in fact develops perfectly normal after this imaging. And the process, this is a calcium imaging process, which is similar to one that we heard earlier on. So this is a transgenic zebrafish, not to be confused with a transgender zebrafish. That's something completely different.
That's tomorrow night.
So a transgenic zebrafish is one that's been genetically modified so that its neurons will express this dye so that they fluoresce when they're active, and it's to do with the calcium chemistry that's going on. So when they're active, then they will fluoresce. So you shine light on this unfortunately, zebrafish larvae are transparent, which makes life a whole lot easier. And you shine light on the brain, and when the neuron is active, then it fluoresces, and then you can image it like this. And so what you can see is these exquisitely beautiful firing patterns going on in the brain. This is somewhat speeded up, but you can in fact see the oscillations that Doug was talking about there. There are oscillations between the forebrain and the hindbrain that show up when you start to analyze what's going on there. So that's the kind of thing that we'd like to be able to simulate on a computer. So how would we do it? Well, so suppose that we were able to construct a kind of map of the connections by using the sort of kind of technology that we were discussing earlier on, and suppose that we were also able to understand how each individual neuron functioned, then sure, the brain is not like a computer, but we can make a computer that's like the brain. So we can start to simulate all of that. We can start to simulate exactly how these neurons behave and how they communicate with each other. So what we're seeing here is it's just 1,000 simulated neurons. And each of these neurons, you're simulating the mathematics involved in transmitting the electrical spikes around that we were talking about earlier on. And you can simulate that on the computer. You can simulate exactly what each neuron is doing as the potential in the membrane builds up, and then it sends out a spike. So the little red dots that you see flying across are when the neuron fires and sends out a spike to all of the neurons that it's connected to. Of course, this is just an animation, and it's an animation of the process that's going on inside the computer. And here you're just seeing 1,000 neurons being simulated, but you can already see that there are similar kinds of complex patterns that you saw in the zebrafish. Although we are talking about very different temporal scales. This is just 1 second that you're seeing here, and whereas you saw many seconds speeded up in the case of the zebrafish. So we can do all of that. We can do all of that kind of thing, and really the enormous challenge for us in trying to build a model of the brain, one of the enormous challenges is really scaling up. Because as you've seen, we need to do all this kind of thing on a truly enormous scale. And scaling up to the human brain is a really enormous challenge. But if we wanted to scale up to, say, first of all, to the brain of, say, a mouse, then maybe that's something that's feasible within 10 years or so. Then what we want to do is build the kind of thing that you've seen just now, but very, very much scaled up.
And this idea of a brain in a vat is wrong-headed, right? There's motion that is necessary.
Yeah, so I think so everything that we've seen so far is a little bit like a brain in the vat. We're seeing disembodied simulations. We saw a disembodied simulation there, and we saw the brain of the zebrafish larva. We just saw the brain isolated from, well, it's not taken out of the body, I'm glad to say. The zebrafish's larva is still there, but we only saw the brain, and we didn't see the body. Whereas brains evolved in order to make bodies move around in the world, the body of the animal move around in the world and interact with the objects in that world in order to improve its chances of survival and reproduction. And that's really fundamentally what brains are for. So if we try and simulate the brain and try and understand it, and we try and build models of it in this disembodied way, I think that's the wrong way to go. So from the perspective of a computer scientist and a roboticist, what we need to do is not just build a computer model of the brain, but we need to embody that computer model. So effectively that means trying to build robots and make your models control robots and make robots move around in the world and interact with things. And we'll see an example of that in the next video, which is due to Tony Prescott from the University of Sheffield and colleagues from the University of Bristol. Now this robot, which is called Shrewbot, now you can see that this robot, which is slightly scary, has got whiskers, and it's the latest in a generation of a series of robots that were produced by this group where they were studying whiskers. So they had a kind of rat bot before this. They discovered some very interesting things. So they were interested in the rat barrel cortex. So that's a little bit of the rat's cortex, which is to do with processing the sensory information that comes in from whiskers. And what they really quite convincingly showed was that you cannot understand a bit of rat barrel cortex by studying it in vitro, studying it in isolation from the signals that are coming in from these actual whiskers, and in the context of a big sensory motor loop whereby the whiskers touch things, and then the sensory information comes from the whiskers as they brush against things, and then that causes the rat to change the way it's whisking. So if it touches something, so if the rat is whisking away like this, and it touches something, was that a good impersonation of a rat whisking? And it touches something on this side of its body, then it will change the way it's whisking on that side of the body in order to try and sense more accurately exactly what's going on there. And that's all part of the big sensory motor circuit, and in order to really understand what all of those nerves, what all those neurons are doing in a context like that, or any context, you've really got to understand that whole sensory motor loop. So if we're going to try and build computer models of the brain, then I think we really need to embody them in robots.
Right, so we started with, we need a lot bigger, better computers. We got the bag of water idea. Now we have animation showing that you argue that it can't just sit there. We need all of this to come together onto an actual object in motion that can think and walk around New York City.
Yeah, and it has to be really, really big. Yeah.
It has to be that big.
Because otherwise it can't climb up the Empire State Building.
Think about it. So, but Greg, jump in on this. I mean, how far are we just from, as we add to our laundry list of impossible tasks, from just creating a robot that can walk like a human?
Well,
or like Doug.
I'm not sure about the pistol.
Who's stubbing his toe.
We're making progress. A bit of potted history before we cue the next video, because it may tie together some of the strands. I should maybe say some of my motivation. What really fascinates me about this topic and others are how well do our mathematical models, do our tools that we have to study a phenomena like the mind or like the brain, how well do they fit with the actual fact of the matter? And this rough interplay that's touched upon, both Doug mentioned that maybe the computer is not the right tool for modeling the mind. If we were to buy it at a computer store, you'd send it back, right? It's very noisy, it's very slow, so maybe that might not be the right model. And likewise, we saw earlier about the complexity of these things. So with this background in mind, take a task that our robot might like to, the capability to walk, yes? In AI, you could look at the history of this, of the walking problem, speech, the main AI problems, reasoning, language understanding. And the first chapter of this, it was, we're going to throw as much computation at this problem as we can. It's deliberation. We're going to prove a solution to this. And this first video, which is only from 2007, instantiates this design philosophy. So this is the first maiden walk of this robot. And the idea is that this thing is really deliberating. It's left, and then it's correcting itself, and then right. It's lurching. It's not at all how we walk. And as you can see, in the end, he falls over. I might add that there were 2 other videos we didn't get the license for them, so I have to describe the trajectory of the point I wanted to make with this. The first is that you can think of this as an act of deliberation. It really is emphasizing the cognitive activity. I'm going to put my left foot forward, and then I'm falling over. Where do I need to put my right foot? And so on until events catch up to him. If you think about how we walk, it's more dance than deliberation. Our hips sway, our arms swing. It's a controlled fall, yes. We pitch ourselves forward, but it's much constrained by our physiology. And indeed, one of the videos I wanted to show you is based on a patent application from 1912 of a toy. This is in the video. If you want to look for it online, it's from Cornell's lab. It's Martin Weiss's work. And all it was is just a mechanical toy that affected walking purely by gravity. So you set this toy on an incline, and the legs and the little arms move as you would expect. There's no motorized mechanism at all. It's all gravity. So the point, we have this computer in the first slide that we saw, really deliberating, left, right, and then you see this very elegant toy. There's no computation at all. It's just done by the physiology. Granted, you need a ramp for the thing to go down and to get it started. And it was this idea that was filed in London, actually, in 1912, based on these toy designs. So the most recent robotics for the walking problem have married these 2 things. Right? So yes, we need to have a controller that's moving the legs and having an intention to walk, but also designing the robot itself to help it along, to put constraints on the problem. And that's the key insight that we've learned throughout AI. If we've learned anything from AI, is that you can't throw more computation at these problems. They're too big. They're just vastly, the search space of trying to find the right solution, to try to find where to put the left foot. God, I was successful. Where now do I put my right foot? That's just not, you're not going to get very far, as we saw in the video. What we need is to find constraints on a problem. It could be language understanding, it could be walking, and I've given a sketch of how that works. There's a gyroscope, basically, and we lean forward and we start swinging our way, and our legs are likely to land more or less where they need to be to keep us from falling over. And we accelerate by leaning forward, yes? So what I'm interested in, and I think is necessary to make progress on this problem, is to find constraints on these problems, because they're just too big to look through the possible solution space for the higher order functions that we want, whether it's walking in this case, or whether it's language, communicating, having beliefs, desires, and so on.
So it has to, as we look at our continued science project here, it has to have a reason to walk. It has to have a reason to move, right? Is that?
Yes. So to tie this back in, we're adding more things to our robot. So I agree that we need to have our mind embodied and moving around in the environment. But I would also add that a robot has to do something. It has to have a purpose. I rather like this one, actually.
That's a
nice
smart
touch.
robot.
But it needs to interact with an environment, have goals, desires. And I might even add that if you're thinking of us as a model, it may need other robots similarly composed. One of the main cognitive capacities that we have, core cognitive capacities, is imitation. We rip off stuff from one another all the time. Social cues, behavioral cues, and learning how motor functions. As children, you can watch them struggling with something on their own, and if a sibling comes along that's mastered it, it accelerates rather quickly. So imitation is something that seems to be fundamental to our brain development and cognitive development. So we may need other robots running around doing things as well.
And you specialize in reason, you think about these things, and talk about decision making in all of this, how that can be measured, and to catch a fly ball, I referenced that at the top, just the amazing computations that go into something like that.
Yeah, so this is a nice example on its own, or I like it anyway. It might help as a simplifying metaphor for some of the challenges that we have, at least this theme that I'm pushing, that we need some way to deal with this complexity. So take the task, and I'm taking this example from Gerd Gigerenzer at Max Planck in Berlin. There are two ideas to take the task of catching a fly ball. Yes? So one idea is to, and this is Dawkins and other psychologists have forwarded this idea. And the idea is basically the player calculates the trajectory of the ball. So it's going to know where the ball goes and then runs to catch it. So this is Dawkins. When a man throws a ball high in the air and catches it again, he behaves as if he'd solved a set of differential equations, predicting the trajectory of the ball. At some subconscious level, something functionally equivalent to the mathematical calculation is going on. So this line of thought is embracing the complexity of this problem. What are we doing? We're doing calculus. We just don't know it. So what else could it be? Well, there's another option. Hold on a minute before giving this slide, because I want to just describe it simply. So the first is to say that we're doing mathematics, even subconsciously, and solving differential equations to catch a ball. That's what all baseball players are doing. The other is something called the gaze heuristic. So the nice idea here is that it simplifies the task, behaves completely different than what I've just described. In fact, the cues that it uses, not a single variable that I wanted them to put up actually the differential equations to show you, but they wouldn't do it. But you have to imagine it. If you haven't calculated a trajectory, you should take a look. It's a little nasty. None of the terms that you would have in the equation are used in the next rule that I'm going to give you, and it happens to turn out that this is what players actually use that are measured. They can't describe it, but they nevertheless follow this pattern. It's 3 rules. And this one works actually when the ball's already aloft. First, fix your gaze on the ball. 2, start running. 3, adjust your speed so that the gaze of the ball, your angle of gaze, stays constant. And if you do this, you'll meet the ball. If you play frisbee with a dog, you'll notice that they're not doing calculus. Often enough, you throw the frisbee, they're not running after it, they're running alongside of it so they can catch the gaze and then jump and catch the ball. You see this with fish, you see this with other animals trying to catch prey or a mate. This can be similar. The idea here is this thing can be used deliberately. This is an intuitive rule. And it's something that was used recently for an event here, this Miracle on the Hudson. Right? You remember this was the plane that took off from LaGuardia, lost both of its engines, and the decision needed to be made quickly whether to return to LaGuardia to land or do something more risky, like land in the Hudson. And basically, it was a version of this gaze heuristic. The co-pilot, Jeffrey Skillies, spotted in the windshield the control tower back at LaGuardia and noticed that the tower was moving ever so slightly up in the windshield, which that is the angle the gaze was changing. And the direction of the change was such that they wouldn't make it. So the decision was made. Well, you know what happened. And this is, interviewed afterwards, this is exactly what Skillies said. So it's not so much a mathematical calculation as visual, in that when you fly an airplane, a point that you can't reach will actually rise your windshield, a point that you are going to overfly will descend in the windshield. Now, why do I raise these examples? there are beautiful, that I study, I love mathematical theories for decision making, for example. And they're very similar to the Dawkins approach about how one ought to reason, norms of reason follow from these things. And then you look to see how people are actually doing this, and it doesn't resemble it at all. So there's this question of, how do you bridge this gap? And what we're hearing on the panel tonight are clues that I, as a theoretician, find tantalizing. So for instance, I'm learning forgetting, for instance, wiping out information is crucial to us. We're bombarded with information, so we have mechanisms to wipe out things so we can focus on the bits that are most salient, that are most relevant to us making a good, sound decision. And likewise, these simple rules that I just mentioned before are exploiting both cognitive capacities that we have and also the interaction that we have with the environment.
You reminded me talking about this. I did a story actually last year at the Mayo Clinic on pluripotent stem cell research. They actually dug a chunk of my arm and grew cardiac tissue out of it. And at one point, the doctor pulled out a slide, and it was they were growing neurons in the same sort of way. And he says, sometimes I look through my microscope and I say, I wonder what that guy's thinking. Do you think in the theoretical, philosophical terms when you're looking at neurons or you're looking at glia at that level? Do you try to make the connections to figure out how this gives us this sense of cognitive awareness?
Well, the sense of wonder, is something we delight in as scientists every day. Cognitive awareness, unconscious mind. Let me ask you, Bill, have you ever been to Moapa, Maryland?
No.
How do you know?
Whoa.
You instantly knew. You didn't go down.
Well, it could have been Heineken, but maybe.
So this is the mysterious part of the brain, the way that it works unconsciously and has access to retrieve information. We seem to have a construct of the universe. You know where you've been, where you haven't been. You didn't run down a list of connections of all the places you've been and check them all off the way you would in a computer. So that's very interesting. The other thing that, in terms of sense of wonder, the point you made about the body is very, very good. Because a lot of what we take for granted as being in the brain actually is in the place of the body. So I'd like everyone in the audience to imagine the last time you were terrified. For me, it's stage fright. So, what did it feel like? Heart doing this, and your respiration's going crazy, and your stomach, and you have to go to the bathroom, and your throat is dry.
For me, it was when you got out that water pistol.
Yeah. And the point is, what if none of those things happened? What if you were just as calm in the same situation, about to be mugged? Would you still be afraid? Can fear exist in the mind? And so I think the body is very important to cognitive function. And in fact, all of those things that I just mentioned have nothing to do with the brain, really. All the neurons are scattered throughout the body. It's the sympathetic nervous system that controls the heart and all these other things.
Right. Murray, let me ask you about the Terminator Matrix question. Say this is possible. We can build a brain. Will it be conscious? Will it be self-aware?
Well, that's different from the Terminator Matrix question. So there's two questions there. So can I do both of them?
Do them.
Okay. Okay. Let's deal with the easier one first, the Terminator question. So I guess there are two motivations why we might be interested in building simulations of the brain. One, of course, is the scientific motivation, that we want to try and understand it and understand how it works. And the idea really is that if we properly understand how it works, then we ought to be able to simulate it on a computer. We ought to be able to build computer models of it. Moreover, if we build computer models, then that will help us to understand it. So it sort of goes both ways. That's one motivation. And then the other motivation is that is a kind of AI motivation, an artificial intelligence motivation, which is that maybe by building models of the brain and understanding better how the brain works, then we might be in a position to build artifacts that exhibit intelligence as we do, or maybe even intelligence as a rat does. Rats can do pretty clever things, and or dogs, and it might be quite handy to have robots with the intelligence of a rat or a dog, which we don't have today. So there are those two motivations. But so if we really could do this, maybe it might be in 20 years' time, or 50 years' time, or even suppose it's in 100 years' time. 100 years' time is going to be within the lifetime of my grandchildren. Which I don't have any grandchildren yet, but if I do, then I potentially care about this, even though it seems like a long time away. So there's a question about what are the ramifications of that for society, and there is one very unpleasant scenario, the kind of Terminator scenario, which I think is actually worth taking seriously. Although I loathe calling it the Terminator scenario, but it's worth taking seriously, and this is one. I'll tell you one reason it's worth taking it seriously, because if we can build something that has the intelligence and cognitive capacities of a chimpanzee or a mouse, or then a chimpanzee and then a human, then I think it's actually inevitable that we'll be able to build something, or that will exceed a human being in its cognitive capacities. Now, it may sound like science fiction, and it is science fiction, but I think we need to actually start asking these kinds of questions now, because we're talking about things that, as I say, may happen within our grandchildren's lifetimes. Moreover, there's a certain argument that goes that if you can build something, an artifact that has human-level intelligence, then it can start to work on its own successors, and you get a kind of speed up of self-improvement, because if it can build something better, then that better thing will be better at building better things, and you get this kind of intelligence explosion that way. And the concern is that how do you really control exactly what the actions of these things that you've built are? It's very difficult to work out how we could really control those kinds of artifacts. So I have to say that I used to just be very dismissive of these kinds of scenarios and these kinds of worries, especially when I was a young AI researcher, and I just wanted to just build these things. And but I'm starting to take them a lot more seriously as the technology is improving. So that was the one question. Can I answer the other one as well?
So go ahead, keep going.
But it's of course a completely different question from the question of consciousness, because maybe you can build something that exhibits the behavior of a very smart human being, perhaps a psychopathic but smart human being, and perhaps it's not going to be conscious at all. Certainly, there are different kinds of ways we might build it. If we build it in a way where we're emulating the way the brain works, then we've got extra reasons for thinking that maybe it's right to ascribe consciousness to it. But we might depart from the way biological brains work and build something that's very alien, and then we might have less reason to think that we could call it conscious. So there are separate kind of questions. You can have something that behaves in a very clever way, but is not conscious at all. That is to say, doesn't have any feelings. It doesn't experience the world in the way we experience the world. It's not capable of suffering or of empathy. So we can imagine all of that, and yet is still capable of, doing all the things that Arnold Schwarzenegger does in the films.
I'm picturing a TV news executive as you describe all of those changes. Please.
So that's a separate question of, would these things be conscious? So let's imagine that we build a perfect simulation of a mouse, say. That's my favorite example, because it's a much more technologically feasible example than a whole human brain, a lot fewer neurons. So suppose that we build a perfect simulation of the brain of a mouse, and we embody it in some kind of furry, fluffy robot. So you'd want it to look realistic, and you wouldn't want a kind of like, a sort of hard metallic body. And it behaved exactly like a mouse. Perhaps you had a particular, original exemplar mouse, Mickey, and it behaves exactly like Mickey. It shows that it's learned the things that Mickey learned during Mickey's lifetime and so on. Then if we think that we have some kind of duty to the lab mouse to prevent it from experiencing suffering, we don't want to torture the lab mouse, well then there's the question of, do we think about whether it's right to torture this thing that we've made? And it sounds ridiculous. Well, of course, it's just an artifact that we've constructed. Surely it's not capable of experiencing pain, so why should we care about it in that way? But there's an argument which I find very, very powerful, which is that imagine that rather than doing this, making this copy, this simulation, all at once, imagine that we did it one neuron at a time. So instead of building a computer simulation, we took one neuron at a time and we replaced each neuron by an electronic equivalent, or a computer, little tiny computer that's simulating what that neuron does. And suppose one by one we replaced all of the neurons in the mouse's brain until eventually we've got something that is completely computer driven, completely electronic. There's nothing biological there anymore in terms of the brain. Well, what happens to the consciousness of that mouse? What happens to its capacity to experience pain? Well, it seems that there are only 3 scenarios that you can really entertain. Maybe its consciousness, you get to a certain point, you get to neuron 125,376, and you replace that one, and consciousness goes, right? That's there's a kind of threshold. Or maybe consciousness just disappears altogether right away as soon as you get rid of the first neuron. Or maybe the consciousness persists throughout. And to my mind, that last scenario seems to be the only one that really, really convinces me. And that argument, for me, seems like quite a persuasive argument for why we should, if we imagine these things not too far in the future, we really should take seriously the idea that they would be capable of consciousness and of suffering.
Greg, what are your thoughts on this?
2 remarks on risk. One is that the argument, this is the self-improving AI, and we should perhaps be worried about this. Those arguments do depend upon disembodied AI. So I haven't seen anything that
Well, why do they depend on disembodied
all
AI?
the arguments that I've seen assume disembodied AI. They're not at all considering power source. They're not how the thing would replicate itself. They're pure thought experiments about the kind of AI that we dismissed earlier in the program. Second thing about risk, it's a more general one, and it's one. I think it's something that just applies to dual-use technology in general. It's a bit more pernicious in the form of such as the technologies and sciences that we're talking about here, but also just information technologies in general, is that we often will develop a science, a technique, a method, a technology with a particular purpose in mind, but it can easily be reused for something else, good, ill. And this is something that we've been dealing with for a very long time. One of the issues that maybe is a bit more pointed, and I don't want to raise this just to dismiss it, is that particularly in the information, the techniques to deal with these large data sets that we were talking about earlier, this is the mother of all dual-use issues, and it's something we need to be aware of in moving forward. I think it's something to well, I don't have concrete thoughts about what one ought to do or one ought not to do with respect to this. I'm just mentioning these dual use or multiple uses of technology are particularly pernicious, or can be particularly pernicious in this domain, because we're looking at so many areas of development. Consciousness, I will punt. I don't think we're anywhere close to, we just don't know enough to begin to, for me anyway, to engage with those thought experiments.
Well, thank you, panel. We're going to take questions now. We've got a couple microphones, I think, set up. And as people who have pressing queries for these brilliant folks, as you line up, we're going to start with a question from our audience in Manila at the Mind Museum there. And this is great. Human brains are famously error-prone. So if we were to build artificial brains, will we have to program these tendencies to make mistakes? It reminds me of the car companies in China stealing American designs and actually engineering the flaws in, right?
Right.
What do you think, Kristen?
Well, I think it's in part a byproduct of something that's very necessary that was mentioned earlier, and that is that you don't want to remember everything that you're sensing, or you will fill up and not be able to manage the world. So not only do you forget on a very rapid basis, you have to pick and choose what's going to be remembered. And some of the things you don't get to choose, right? So post-traumatic stress syndrome will evolve if there's memories that are traumatic. It takes a long time to forget them and unlearn them. But there are many things that just happened tonight that you're not going to remember. And so the errors are sometimes simply a byproduct of something that the brain has to do, and that is be able to also filter and to forget. And so
weren't you saying that the recent research about sleep erasing, like wiping hard drives as you rest?
A memory is not a recording. It has to have a schema. It has to have context.
Oh, he's got a brush now.
I'm worried of these props.
But think about it.
He's the carrot top of brain science.
This is because all our ideas about learning and memory go back to Pavlov, coincident activation, sensory input. But making a synapse stronger is not a memory. There's things like novelty. Because memory is about the future, what's likely to be important, and something that's novel, out of context, will be remembered. So what happens is if you remember anything
remember that.
tonight, it will be, if you remember anything from this, it will be because tonight, when you're asleep, you'll replay the events during the day, and you'll associate it with emotions and information you already have in your brain to relate to that, and you're going to throw out what's not important, most of it, and you're going to then associate. Those neurons aren't wired together, so what was novel? How did you feel about it? What was the time? What was the context? What were all the multiple senses that went into this memory? So our research, recent research has shown that also the old our idea about how neurons work goes back to the neuron doctrine, that the impulse goes into the dendrite and out the axon. Well, during slow wave sleep, the brain and the hippocampus undergoes high frequency oscillations, and the neurons fire backwards. So, and our lab has recently shown that erases memories, now, which is very important, as you mentioned. So, but the point I'd like to make here is that intrinsic activity that goes on in the brain as opposed to sensory evoked activity, is very different than sensory evoked activity. And when we're trying to understand how the brain works, the fact is we don't understand the language of the brain. Is it a frequency code? Is it brain waves? It's a little bit of all these things. So and my own interest is in exploring beyond the neuron doctrine and trying to find new ways of thinking about how the brain works.
Okay. Let's take some questions. Why don't we start on this side? What's your name? Hey, Alex.
I kind of want to touch a little bit into the effort and the billion-dollar challenge to map the brain, and touching a little bit in with what you said about constraints. Do you feel that having that challenge is kind of leading the effort in a direction and putting a lot of focus in just that one direction, versus if you didn't have that challenge and you were forced to do it a different way, like looking for patterns or any other kind of way to solve this problem?
Just to me?
For anybody, I guess. Maybe Kristen.
I can offer some thoughts about that. So despite the fact that there appears to be a white paper that what's going to happen is we're going to map where every single activity occurs in the brain and be able to have a brain map of activity, implicit in all of that is way beyond just the activity of individual neurons. It really is all the kinds of mapping that we've talked about. And while the amount of money that's been put into it at this point is certainly not enough to do all of what we've been talking about, by focusing some of the energy and the effort on trying to think about, well, what should we prioritize, and how can we bring together different fields to understand the brain, people who haven't been thinking about developing tools to understand the brain better, I think there's going to be a lot of synergy that comes out of it, rather than, that there's a threat to some particular field of not being able to get enough resources to keep going forward in the brain. So I really think that the future is very bright when you have a president and you have, the European community starting to say, look, we are starting to get to some thresholds where we're going to be able to learn something about the brain if we can bring all of the great minds together to look at new ways to investigate the brain and try to get this trajectory off the slope that it's on now and make it much faster.
Thanks.
Okay, over here.
Hi, my name is Samai. Thank you for being here. And can you give us a little insight on the manifestation of synesthesia and your opinion on whether you think it's a advantageous evolution of the human mind?
Seeing music or
Yes.
Well, seeing numbers as colors
Yeah.
and things of that sort. I don't know enough
Anybody
about it.
thoughts on that?
Well,
Doug may know
there's a
more.
big question as to why we don't have synesthesia, or it's called the binding problem. The neurons that look at the color, that analyze the color red on your shirt, are not the same neurons that handle shape, that handle motion. So what keeps the red on your shirt when you walk around this room? So this gets really into some long-standing mysteries in the brain as to how information that's being analyzed in different parts of the brain gets put together into one object. My arm moves here. Why is it all perceived as part of my body? So that's a bit of synesthesia, is when that process goes wrong, that different senses become partitioned incorrectly. So, I don't have an answer, but I think that gets at the question that we're talking about.
Young man, what's your name?
Callum.
Hi, Alan.
Okay. A few weeks ago, I got this subscription email from it was either TED or Nature magazine that it talked about this technique for making brain tissue clear or something. I didn't exactly have time to read it all, but I was wondering if there was a way, like, if you could just individually label the the axons in the brain, then the glia cells in the brain, then the synapses, and then somehow do a 3D scanning of that. Is that how you're approaching it, or?
That is an approach that is being used now to be able to colorize different circuits, that you know something about the gene that you've put into that circuit, and to be able to follow. The clarity aspect of it is that because of all that myelin that's white and gooey, it's very hard when you label something to actually see where it went in the whole brain. But if you can substitute that, the fat in your brain, with something that's clear and still keep the neurons kind of intact, and for them to still hold their dye, then you can actually see them without having to cut up the brain and do the kinds of reconstructions. You won't be able to do the kinds of reconstructions I showed you because clarity is a light microscopy technique at this point in time. But one of the big questions that actually is being pursued is to see whether or not, when you substitute the fat in the brain with something that's clear, and I can't remember the exact name of the molecule that they're using, but it's actually, it substitutes for those that are making it impossible for us to see through the brain, to see whether or not, if you section it in the way I have for the electron microscope, we can actually get down to the nanoscale analysis. And the answer is not in yet, but boy, are you on target with the latest, hottest technique in neuroscience.
Thanks, Alan. Yes, sir.
Hi, my name is Tyler, and I work in a research laboratory, but one thing that's interested me is an analytical tradition in philosophy of mind where you have people like the functionalists on one hand, like Daniel Dennett, arguing that you don't need to understand the brain to really understand the mind. You can build a mind out of any substance. Whereas you have John Searle, on the other hand, who would argue that you need to understand the brain-mind identity as sort of a single unit. So given that we don't really know where the unit of information is currently, whether it's individual neurons or population codes, et cetera, how close do you think we are to resolving that sort of debate? Or of how close you need to model the brain to model the mind?
That's yours, yeah.
Right.
Yeah, so I mean, it's interesting that you raise this, because I started out trying to do artificial intelligence in a very, very different way, by engineering AI from scratch, trying to build logic-based representations and explicit reasoning mechanisms and so on. And I kind of eventually felt that this was going nowhere in terms of building, real kind of human level AI. So at that point, I turned to the one exemplar we have of something that really works, which is not just my brain, but our brains, biological brains. And because if we can understand them, then I think we're really getting somewhere. Now that doesn't necessarily. So obviously, if we do understand how biological brains produce cognition and consciousness, then we've taken great strides to understanding cognition and consciousness. But that doesn't necessarily mean that that's the only way to do it, though. So maybe having achieved that, if we can achieve that one day, and we do, as Kristen said earlier on, abstract out certain principles of how this works, then we might find that there are deep principles that are analogous to the physical principles of aerodynamics that govern both natural flight and fixed-wing aircraft, and we can come back and we could engineer something in a completely different way. So that would support that. Maybe that's how things will turn out, and that supports, I think, a kind of broadly functionalist way of looking at things, if that turns out to be the case.
I'm rather interested in the ancillary questions that will likely emerge as we make progress. I'll give you an historical analogy, because we're not in a position to identify what those would be. Take Descartes, right, and his problem of skepticism. Really worried about having a system whereby I have a belief that has enough justification that I can justify the beliefs I have about the external world. Cogito ergo sum, yeah? And well, one of these, that problem for some is still open, the problem of skepticism. But there are other issues that I would offer as very serious. This predated statistics, how to measure, how to control error, how to know when we're wrong. And the worries that Descartes was articulating, well, there's a subset of questions that broke off, and there's now a science that's developed, and we're corralling these, yes? So my guess is that these large, big questions, they're interesting, very important, but for me, I like these. I'm really excited about these second order, or the second level questions of these kinds, and I think there's going to be a lot of them that are going to be peeled off and give us tremendous insight into who we are, without perhaps resolving this dispute between Dennett and Searle.
Well, thank you all. I'm afraid the folks in line, we're out of time. The clock has been on zero. We're about 5 minutes over. But please, for Doug Fields and Kristen Harris and Murray Shanahan and Greg Wheeler, give them a big round of applause. It was humbling and thrilling to share the stage with these folks. And I should mention before you leave, if this topic interests you further, there is a salon tomorrow at 1 o'clock, all-star in-depth conversation about this whole initiative, both here in the States and in Europe. And you can find more information on worldsciencefestival.com. Thanks for coming, everybody. We'll see you next time.
It's fun.
Any of those questions?
It was a lot of fun.
Thank you very much.